Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
The genetic control of early embryonic development, recognized by the 1995 Nobel Prize awarded to Edward B. Lewis, Christiane Nüsslein-Volhard, and Eric Wieschaus, stands as a monumental achievement in the history of biology. Its genesis lies in a confluence of factors: the maturation of genetics as a discipline, the emergence of powerful molecular techniques, and the persistent quest to understand the fundamental mechanisms underlying the remarkable process of embryogenesis. To fully appreciate the significance of this work, one must first consider its intellectual antecedents. Prior to the discoveries of Lewis, Nüsslein-Volhard, and Wieschaus, embryological research was largely descriptive. Scientists meticulously documented the stages of development in various organisms, carefully observing the morphological changes that transformed a single fertilized egg into a complex, multicellular organism. However, the underlying principles that governed these dramatic transformations remained elusive. Wilhelm Roe's experiments in the late 19th century, for instance, although groundbreaking, were based on a proformationist viewpoint, indicating that the fertilized egg already contained a miniature version of the adult organism, which merely unfolded during development. This view was later challenged by Hans Triesse, whose experiments on sea urchin embryos demonstrated that earlier embryonic cells were totipotent, capable of developing into a complete organism under the right conditions. This supported the idea that cell fate was determined through interactions among cells, rather than being predetermined from this. The development of genetics in the early 20th century provided a framework for understanding heredity and variation, but its connection to embryology remained tenuous. The rediscovery of Mendel's laws and the subsequent mapping of genes onto chromosomes offered a powerful tool for dissecting biological processes, yet the molecular nature of genes and their role in development remained obscure. Thomas Hunt Morgan's work on Drosophila melanogaster, the common fruit fly, proved invaluable in laying the foundations of modern genetics. Morgan and his colleagues meticulously studied mutants with altered body plans, demonstrating that genes could control specific aspects of development. However, they were unable to identify the genes themselves or elucidate their mechanisms of action. The advent of molecular biology in the mid-20th century revolutionized the study of genetics and embryology. The discovery of DNA as the carrier of genetic information and the elucidation of the genetic code provided a powerful framework for understanding how genes control cellular processes. The development of recombinant DNA technology allows scientists to isolate and manipulate genes, opening up new avenues for study and development at a molecular level. Edward B. Lewis, starting in the 1940s, made pivotal observations concerning the bithorax complex in Drosophila. His painstaking genetic analysis revealed that these genes were arranged on the chromosome in the same order as the body segments. They influenced flies with mutations in these genes displayed bizarre transformations of body parts, such as the development of a second pair of wings where the halters, balancing organs, should have been. This observation led Lewis to propose the concept of homeotic genes, genes that specify the identity of body segments. Christiane Nüsslein-Volhard and Eric Wieschaus, building upon Lewis' work and the advances in molecular genetics, embarked on a larger-scale mutagenesis screen in Drosophila to identify all of the genes required for earlier embryonic development. They reasoned that mutations in essential developmental genes would result in lethal phenotypes, preventing embryos from developing normally. Through meticulous screening of thousands of mutagenized fly line, they identified a selection of genes that were crucial for establishing the body plan of the developing embryo. Their research, conducted primarily across the late 1970s and early 1980s, involved inducing random mutations in Drosophila, then carefully examining first-generation larvae for anatomical abnormalities. They categorized these mutations based upon the types of defects they produced. One set of mutations affected segment polarity, leading to embryos with mirror image duplications of segments. Another set interfered with the total number of segments, generating larvae with larger, fewer segments or smaller, more numerous ones. Gap genes, parallel genes, and segment polarity genes were identified in this manner, each playing a distinct role in the hierarchical process of establishing the body plan. The power of their approach lay in its comprehensiveness. By systematically screening for all possible mutations affecting early development, they were able to identify a nearly complete set of genes required for this process. Moreover, their genetic analysis allowed them to establish the hierarchical relationships among these genes. For example, they found that gap genes, such as hunchback and crapelle, are expressed in broad domains along the anterior-posterior axis of the embryo, and that these genes regulate the expression of parallel genes, such as even-skipped and Fushitarazu, which are expressed in alternating stripes. In turn, the parallel genes regulate the expression of segment polarity genes, such as engrailed and wingless, which are expressed in specific compartments within each segment. Nüsslein-Volhard and Wieschaus' work also revealed the importance of maternal effect genes. These genes are expressed by the mother during oogenesis, and their products are deposited in the egg. The products of maternal effect genes, such as bicoid and nano-S, establish gradients of morphogens, signaling molecules that specify cell fate based on their concentration. Bicoid, for example, is a transcription factor that is concentrated at the anterior end of the embryo and activates the expression of genes required for head and thorax development. Nano-S, on the other hand, concentrated at the posterior end of the embryo and represses the expression of genes required for abdomen development. The immediate impact of the work by Lewis, Nüsslein-Volhard, and Wieschaus was profound. It provided a detailed genetic blueprint for early embryonic development in Drosophila and established a powerful framework for studying development in other organisms. The identification of homeotic genes and the discovery of their conserved role in development across diverse species revolutionized our understanding of evolution. The discovery that similar genes control development in organisms as different as flies and humans suggested that the evolutionary history of development was deeply conserved. Subsequent research revealed that homeotic genes, now known as Hox genes in vertebrates, are arranged on chromosomes in the same order as the body regions they specify, mirroring Lewis' initial observations in Drosophila. Moreover, the Hox genes in vertebrates are regulated by similar signaling pathways as the gap genes and parallel genes in Drosophila. This remarkable conservation of developmental mechanisms across diverse species provided strong evidence for the evolutionary connections between organisms and highlighted the power of studying model organisms like Drosophila to understand fundamental biological processes. The work of Lewis, Nüsslein-Volhard, and Wieschaus profoundly influenced subsequent research in developmental biology. Their discoveries spawned a vast body of research aimed at understanding the molecular mechanisms by which these genes control development. This research led to the identification of numerous signaling pathways, transcription factors, and other molecules that play crucial roles in embryonic development. For example, the WNT signaling pathway, which is activated by the segment polarity gene wingless in Drosophila, was found to play a critical role in cell proliferation, cell fate determination, and tissue patterning in a wide range of organisms. Similarly, the HOG signaling pathway, which is activated by the segment polarity gene HARP in Drosophila, was found to play a critical role in cell differentiation, tissue morphogenesis, and the development of the nervous system. The discoveries of Lewis, Nüsslein-Volhard, and Wieschaus also had a significant impact on the study of human development and disease. The identification of Hox genes and their role in specifying body plan led to the discovery that mutations in these genes can cause birth defects in humans. For example, mutations in the HOX13 gene can cause sympolydactyly, a condition characterized by the fusion of fingers and toes. Similarly, other developmental genes have been linked to a variety of other birth defects, including cleft palate, heart defects, and neural tube defects. Moreover, the signaling pathways that control embryonic development have also been implicated in cancer. The WNT and HOG signaling pathways, for example, are frequently activated in cancer cells, promoting cell proliferation and inhibiting cell death. Understanding how these pathways are regulated in normal development has provided insights into how they can be targeted for cancer therapy. The reception of Lewis, Nüsslein-Volhard, and Wieschaus' work was overwhelmingly positive. Their discoveries were widely recognized as a major breakthrough in developmental biology, and they were awarded numerous prizes and honors, culminating in the Nobel Prize in Physiology or Medicine in 1995. However, their work was not without its critics. Some scientists question the generality of their findings, arguing that the developmental mechanisms in Drosophila might be unique to insects and not applicable to other organisms. Others argued that their genetic approach was too simplistic and that it failed to capture the complexity of embryonic development. These criticisms, however, proved to be largely unfounded. Subsequent research demonstrated that the developmental mechanisms discovered in Drosophila are indeed highly conserved across diverse species, including humans. Moreover, the genetic approach pioneered by Lewis, Nüsslein-Volhard, and Wieschaus has been successfully applied to the study of development in a wide range of organisms, providing valuable insights into the molecular basis of development. Later revisions and deeper understandings of the work of Lewis, Nüsslein-Volhard, and Wieschaus have focused on the integration of genetics with other disciplines, such as cell biology, biochemistry, and biophysics. These interdisciplinary approaches have provided a more complete picture of the complex processes that govern embryonic development. For example, advances in imaging techniques have allowed scientists to visualize the dynamic interactions among cells and molecules during development in real time. Similarly, advances in genomics and proteomics have allowed scientists to identify and characterize the vast array of genes and proteins that are involved in development. The influence of the 1995 Nobel laureates extends beyond the realm of developmental biology. Their work has inspired researchers in other fields, such as evolutionary biology, systems biology, and regenerative medicine. The discovery of conserved developmental mechanisms across diverse species has provided insights into the evolutionary history of development. Furthermore, the understanding of signaling pathways that control cell fate and tissue patterning is informing the development of new strategies for regenerative medicine, the goal of which is to repair or replace damaged tissues and organs. In summary, the work of Edward B. Lewis, Christiane Nüsslein-Volhard, and Eric Wieschaus on the genetic control of early embryonic development represents a watershed moment in the history of biology. Their discoveries provided a detailed genetic blueprint for earlier embryonic development in Drosophila and established a powerful framework for studying development in other organisms. Their work has had a profound impact on our understanding of development, evolution, human health, and disease. Their legacy continues to inspire researchers in diverse fields to unravel the mysteries of life. It is a testament to the power of curiosity-driven research and the importance of studying seemingly simple organisms to gain insights into fundamental biological processes. The elegance of their genetic screens and the profound implications of their findings serve as a guiding light for generations of scientists to come. The very notion that a simple fruit fly could unlock the secrets of embryonic development across the animal kingdom is an irony that speaks volumes about the interconnectedness of life and the power of scientific inquiry.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.